Good afternoon, ladies and gentlemen. I'm Wei Guo from ProBio mRNA team. I'm so happy to be here to discuss with you today on the topic of integrated RNA-tLNP platform empowering in vivo CAR T therapies. In my presentation today, firstly, I will give a brief introduction of recent progress in the application of RNA-tLNP for in vivo CAR T. Second, I will share some key considerations of RNA-tLNP technology for CAR T therapy. Third, I will introduce the technical challenges and solutions to circular RNA manufacturing according to our circular RNA-tLNP CMC practice. I hope this will be helpful as we work together to explore RNA-tLNP products for in vivo CAR T therapies. In recent years, in vivo CAR T technology has emerged as a powerful new force in the biopharmaceutical field and has rapidly become a central focus of industrial attention. In vivo CAR T therapy offers potential advantages in cost, accessibility, and safety and immediacy. From ex vivo to in vivo CAR T makes CAR T therapy from exclusive to accessible. It enables the technology to be developed as an off-the-shelf product and shortening the waiting time for patients. There are two mainstream approaches to achieving in vivo CAR T therapy: the mRNA targeting LNP and targeting LVV. Both of the pathways have stepped into clinical validation stage in the recent years, such as Capstan, Create Medicine, Unicar Therapy, and Magic RNA. These companies are using mRNA-tLNP technology. Another group of companies, such as Isobiotech, Umoga, Interiors, Genocure, CSPC, these companies using LVV technology to explore in vivo CAR T. In China, investigator-initiated trials, also called IITs, are rapidly accelerating the development of in vivo CAR T therapies, acting as a major hotspot by leveraging over 10 early phase trials, makes this innovative therapy move from concept to clinical. ProBio is an international and professional CDMO company, and we have established both of mRNA-tLNP and LVV platforms, serving CMC studies, clinical product manufacturing, and even commercial product manufacturing. Both of the technology has its own special features. mRNA-tLNP pathways has its transient expression pattern, and it has a rapid PK and a strong profile. It enables repeat dosing with flexible dose adjustment, and it can be cell-free manufactured with short lead times and no production cost. RNA-LNP-based therapies hold great promise for the treatment of B-cell-driven disease and autoimmune disease. Compared with RNA-tLNP pathway, LVV pathway possesses distinct advantages, namely sustained expression, enabling long-term cytotoxicity, and a mature scalable manufacturing process, which makes it more suitable for persistent long-term cancer treatment, such as solid tumor. Today, we will focus on RNA-tLNP technology. Here is a classic picture of RNA-tLNP nanoparticle structure. It features a rich, diverse design, different payloads. It can be linear mRNA, circular RNA, and even proteins, and different targeting binders conjugated on the surface, such as antibody, different kinds of antibodies such as VHH, SCFV, and peptide, and engineered proteins, including new ionizable lipids and spatial structure lipids, and even new formulations are designed to make LNP to be liver detargeting and even spleen targeted. ProBio established a comprehensive platform support client projects from molecular design to IND enabling. And we have two main technology platforms, CGT CDMO platform and antibody CDMO platform. The CGT CDMO platform consists of plasmid, RNA, LNP. We have whole chain RNA-tLNP project delivery experience with 7 plasmid CMC, RNA CMC, LNP CMC, antibody CMC, and RNA-tLNP CMC. And then goes to scale up and quality study, and then enter into the IIT or IND application. And we can provide one-stop solution to in vivo CAR T products. Today, I will introduce the new payload format, circRNA, and why we choose circRNA, what the advantages of circRNA compared to traditional linear mRNA. And circRNA is a single-strand RNA molecules with a covalently closed circular structure. Its lacks of free 5' and 3' ends making it resistant to degradation by exonucleus. And its structural feature grants circRNA a significantly longer half-life compared to linear mRNA, providing a foundation for its long-lasting effects. It can be higher expression, longer half-life, and lead to lower immunogenicity. circRNA drug has moved into clinical validation phase in the recent years, as listed in the table of recent progress of circRNA drug pipelines. circRNA-tLNP products has entered into Chinese IIT and phase 1 in Australia. There are 2 big deals achieved between circRNA Star Company and Big Pharma. BMS acquired privately Orbital Therapeutics for $1.5 billion in cash in October 2025 and recently. Eli Lilly announced to buy Ono Therapeutics for up to $2.4 billion in cash. circRNA has shown great potential as new format payload for application in vivo CAR T therapy. So there is some following questions. How to obtain a molecule suitable for advancement into CMC? What are the critical considerations of circRNA drug development? It can be divided into 4 parts: sequence design, process design and control, scale up, and analytical testing. Sequence is one of the most important parts of circRNA, as sequence is strongly correlated with circRNA yield and product-related impurities like NICT. There are also lots of patterns on circularization design. A well-designed circRNA molecule should result in high circRNA ratio and low leaked RNA. It needs rational process design and control to remove impurities and harvest target product circRNA. Regarding to scale up, mixing, incubation time, compatibility, and turbulence occurred during mixing should take into considerations. At last, analytical technology is critical for circRNA. It requires high resolution and fast testing method to support intermediate process control and product release, especially for the separation of circular RNA and nicked RNA. As circular RNA sequence design is strongly related to circular RNA process and product quality, and how to obtain a well-designed circular RNA sequence. And we need to do functional motif screening, including introns responsible for splicing, IRES, which is strongly related to expression, and do some sequence optimization. ProBio established IRES pool and sequence design platform already, and to help clients to do circular RNA design and achieving higher expression and longer half-life time, and lower immunogenicity and higher circular RNA yield. For sequence design, and we have a well-established one-stop testing platform, and from sequence design to testing, to get a good CMC candidate molecular, including T4 ligase circularization method and intron-based circularization system. ProBio established the proprietary circularization method and patented already, and which could be used for our clients who require it. Our circularization design can be divided into two strategies: regular design and customized design. They are suitable for different scenarios. For regular design, it is suitable for non-coding circular RNA. For customized design, it is suitable for non-coding and coding RNA. And IRES screening and synergetic optimization strategies has been developed in our platform. We established a three-step systematic design, sequence design route, including structure-first strategy and stepwise synergistic optimization, and finally closed-loop validation by experiment. Through IRES screening and RNA sequence optimization, including structural regulation and CAI optimization, the protein expression can be increased by over 20-fold. Through screening of ribosomes and splicing sites, and we can obtain high circularization molecular for further CMC study. The circularization ratio can be up to 60%, and achieving high final product yield is about 5 grams per liter. Here is a case study of circular RNA products from sequence design to manufacturing. Several candidate design to obtain a CMC candidate, and then scale up to get high quality circular RNA products. The standard manufacturing workflow consists of IVT, circularization, TFF, dephosphorylation, TFF, purification, TFF, and filtration. It is more complex than traditional mRNA manufacturing process and be very different. And we have two purification methods for circular RNA: the enzymatic method and the chromatographic purification method. Both of them can achieve high quality circular RNA. Which kind of purification method will be chosen according to the impurities map of circular RNA testing results, and also strong related to cost. In summary, we established comprehensive strategies and solid process platform to solve the challenges existed in CMC study. For example, proprietary circularization method and validated IRES pool for lacking of sequence design capability, and lacking of circularization method. And we have well-established diversified purification platform for high-quality products and better impurity removal effects. Apart from process, platform has been established in ProBio, a well-established analytical platform to support process development and product release. Under the guidance of industrial reference files and ICH, including general properties, strengths, purity, impurities, identity, safety, and potency. The testing items highlighted in orange are circular RNA purity and impurities testing via PA800 or HPLC, which is different from linear mRNA testing. The method should be capable of distinguish linked RNA and circular RNA, which a little difference between linked RNA and circular RNA in structure and molecular weight. ProBio has established the authology circular RNA purity analysis method. We compared 4 different purity analysis methods commonly used in circular RNA purity analysis. Easier is denatured scale can be used for separation of linked RNA, pre-RNA, intron, and circular RNA. It is commonly used in research stage and has been reported frequently. It cannot be used for precise quantification for circular RNA. And C5200. This is a kind of capillary electrophoresis system. It cannot separate linked RNA and circular RNA, and is not recommended for circular RNA purity analysis. And PA800, which is based on the high-resolution capillary electrophoresis. It's a fast method and can be treated as QC method. And HPLC is also recommended for purity analysis, as they can separate linked RNA and circular RNA individually and can be conducted quickly. To sum up briefly, good sequence design and rational process design and control supported by solid analytical platform led to good circular RNA quality and rational process. We announced a white paper on circular RNA in March 2026. It's titled as From Design to Production, Overcoming the Manufacturing Challenges of Circular RNA. Please don't hesitate to log in our website or contact with us to get this material. That's all. Thank you for attention. Hope it will be helpful for us to move circular RNA from research stage to clinical stage. Thank you.